The overall goal of this multidisciplinary project is to isolate the specific mechanisms by which fungicide residue on pollen may cause colony declines in bumblebees.
This method can help answer key questions relating to the high mortality among larval bumblebees as affected by the fungicide-mediated disruption of bee microbe symbioses.
The main advantage of this technique is that it investigates the indirect effects of fungicides on bees by tracking bee demographics alongside shifts in the microbial communities within bee pollen.
Although these methods provide insight into native bee health, it can also be applied to other pollinators, such as honeybees, which play a critical role in crop pollination globally, thereby emphasizing the wider application of fungicides to flowering crops.
Demonstrating the procedures in this collaborative study will be Prarthana Dharampal, Luis Diaz-Garcia, and Caitlin Carlson.
To begin, set up mesh cages in a field planted with oats. Prepare 1 per replicate. Plan to make at least 10 for a single treatment experiment. First, dig a trench around each cage and dig all 4 edges of the mesh cage into the ground to ensure that the bees cannot escape. Then, stock the cages with potted flowering plants that are known to be attractive to bees, such as buckwheat, borage, alyssum, cosmos, and sunflowers. Also, supplement each cage with a single tray of in-bloom clover. Cluster all the floral resources within one corner of the cage, occupying about 2.5 square.